Aloha class, and welcome to my presentation on the plague. I'll give you a brief history. There were three major plague endemics. The first was the Justinian plague, and that took place in 541 AD, and about 25 million people died because of it. The most famous plague, and the one that I'm sure you are all familiar with, was the Great Plague, or Black Death. It originated in China in 1334, and it spread along trade routes to Europe from ships which were carrying infected rodents and fleas. And we all know that it was pretty disastrous, and it killed 60% of the European population. It was also pretty important because it helped Europe bridge into the Renaissance and out of the Dark Ages. The last major plague that happened is called the Modern Plague. It began in China in 1860, and over 20 years it spread from ships from port city to port city, and 10 million people died. Now, the history of the plague in the United States started in 1900. Steamships from Asia brought infested rodents and fleas over, and the U.S. plague endemic lasted from 1924 to 1925. It wasn't very long. And anyone who is familiar with Monty Python will get this. If not, I don't really have time to explain it. So what is the plague? How is it classified? The domain is bacteria because it's unicellular. The kingdom is prokaryote. There's an absence of a nuclear membrane, and the DNA is not organized into chromosomes. The phylum is Proteobacteria. It's gram-negative, and it's a facultative anaerobe, which means it can live without oxygen. The class is Gamma Proteobacteria. It's a purple sulfur bacteria. The family is Enterobacteriaceae because it lacks oxidase and is a non-spore-forming bacteria. The genus is Yersinia, and the species is Yersinia pestis. So, Yersinia pestis, what is it? It's a gram-negative rod, as you can see from the picture. It's a facultative anaerobe, which means it can live without the presence of oxygen. It's non-motile and non-spore forming. The virulence factors. It encodes two antigenic molecules, the first one being the F1 capsular antigen and the second one being the VW antigen. Both of these are needed for pathogenicity, and they are not expressed at temperatures lower than 37 degrees Celsius. As you'll see, the main carrier for Yersinia pestis are fleas from rodents, and the main reason why this doesn't affect fleas is because the flea's body temperature is at 25 degrees Celsius. If you look at the cellular level, if you want to break it down for you nerds, translocation of Yersinia's outer proteins, which are called YOPs, they block the host cell's ability to communicate with the immune system, and they downregulate the response of the phagocytic host cell to infection. And then there's a thing called the type 3 secretion system. Basically, all that does is it injects bacterial proteins into a host cell, and with that, the YOP-H and the protein kinase A are delivered into the host cell, and they subvert signal transduction and inhibit oxidative bursts. The rough, short lipopolysaccharide chains on the outer membrane aid the antibody resistance by causing abnormal attachment of membranes attack complexes, or MACs. And in this next picture, you will see on B, if you look down in the rough LPS, you can see kinda how that would aid in that. Transmission. So, like I was saying earlier, fleas are the main way that this gets transmitted. Fleas from infected rodents. Once one of the infected rodents dies, the fleas will look for a new host to feast on. And anyone who has pets knows how much of a problem that can be. So you and your pet need to avoid contaminated areas where rodents have died. The common area where this happens are semi-arid and forest region. These are areas where rats can thrive. And this is just a basic transmission process. This is basically all you need to know. The animal or rodent or whatever gets infected, the flea bites the animal, and then the flea bites us. It's pretty simple. Diagnosis. So if you were in the hospital and you came in and you thought you had the plague, basically they would collect, definitely sputum and blood, but sometimes a lymph node biopsy is performed, and it's sent to the lab for evaluation. Now, we in the lab, we were able to do qualitative testing, which can give an answer pretty quickly, but to confirm what it actually is, you need 24 to 48 hours to grow it out, which is pretty common with anything in microbiology. This is the plague ecology in the United States. It's pretty simple. As you see in the picture, the fleas are feeding on the squirrels who have the plague, and then the squirrel will die, and then the flea, they just look for whatever they can. So dog, cat, rabbit, us. You need to be very, very, very careful if you have pets and you live in these areas. Animals in the plague. So yes, like I said, the plague, it does affect animals. It won't affect the fleas, but the fleas will carry it to other animals. So dogs, cats, rabbits, any common animal that's kind of in the southwest, of the United States, or semi-arid and forest regions of the world, can all get infected by it. So flea prevention is a must if you live in these areas. Anyone who owns pets knows, how fleas can multiply. It's very, very dangerous. Another way to contract the plague would be coming into contact with the flesh and tissue of dead infected animals. You and I are probably not going to go out and play with dead infected animals, but dogs and cats love to play with that stuff. So it's very important that you keep them away from that. And another way that this is spread is if you're into hunting and you're skinning and cleaning animals. If that animal is infected, it's a very common way to pick up the bubonic or septicemic plague. So the three types of plagues that we need to be concerned with: the bubonic, the septicemic, and the pneumonic. So as you see, the bubonic affects the lymphatic system, the septicemic, the circulatory system, and the pneumonic is when the bacteria gets into your lungs. The bubonic plague. The bubonic plague is caused by Yersinia pestis. The infection is caused when the infected flea or rodent bites the host. And it spreads and multiplies in the body's lymphatic system. And the incubation period for this is 2 to 6 days after being infected. So it's pretty quick. The lymphatic system, it's a major component of the body's immune system. As you know, spleen, thymus, lymph nodes, all kinds of important stuff. Symptoms of the bubonic plague: fever, headache, chills, swollen lymph nodes, or buboes, hence the bubonic name. As you see in the picture, lymph nodes, everybody's kind of familiar where most of those are. The armpit and the neck is very common, but also in the groin, as you see in that picture. It's pretty nasty and painful. Treatment options for the bubonic plague. As soon as it's suspected, you need to get to the doctor, and what they'll do is they'll give you IV antibiotic treatment. In the United States, gentamicin is the most commonly used. Streptomycin, which I know you've probably all heard of, is another very, very, very good antibiotic, but it's just not readily available in the United States. And for all of you nursing students, I also included the dosage information there in case you were interested. If the bubonic plague is not treated right away, the bacteria will spread to other parts of the body, and this often leads to the septicemic plague. So what is the septicemic plague? The septicemic plague, also it's when Yersinia pestis enters the bloodstream of the infected patient. And you know, when bacteria enters the bloodstream, it's a little more serious. And like I said, it manifests from untreated bubonic plague, or can happen from a flea bite and just skip the bubonic plague altogether. The symptoms: fever, chills, abdominal pain, weakness. You know, this is most known for when skin and tissues, they turn black and die. Basically the fingers, toes, and the nose. Hence the Black Death term. Yeah, very, very nasty stuff. The treatment options. It's the same as the bubonic plague. They're going to use IV antibiotic treatment. If this goes left untreated, the mortality rate is really high. It's 50 to 90%. If treated early, that drops to about 15%. So, you know, you need to go get it done right away if it's suspected. The pneumonic plague, the third and final one, is definitely the scariest. It's the most serious form. It's when the infection spreads to the lungs, and it can result from untreated bubonic and septicemic. But it can also happen just from a flea bite or a rodent bite. The transmission. This is the only form of the plague that can spread from human to human, and the reason is because it's an airborne disease. It occurs when a human inhales an infectious droplet. It's very, very scary, very dangerous. The symptoms of the pneumonic plague are fever, chills, weakness, and pneumonia, hence the name. The pneumonia is pretty bad. You have a nasty cough with bloody, watery, or slimy mucus, and very intense chest pain. The problem is when these symptoms get to this point, you're pretty much dead. You know, you only have a few days to live once this stuff gets into your lungs. The treatment options. I mean, like I said, you have to go immediately. You need to get on IV antibiotics if you want a chance to live. So as I said, you know, this one, the pneumonic plague, is very scary, and bioterrorism and chemical warfare. The pneumonic plague is mentioned when talking about that. It's ideal for chemical warfare because it's airborne. You know, once it's in the air, you know, it causes fever, cough, chest pain within 1 to 6 days, and then, you know, in 2 to 4 days you get into septic shock. And brief story: when I was in the Army, I was a lab tech, and this was mentioned pretty high up on the list of bioterrorism weapons that were a threat. So this is actually a really, really high level concern right now. And so they're kind of in the process of developing more vaccines. They're not readily available right now, but they are working on it. So this is a map of the United States on where all the reported cases have happened. And like I said before, semi-arid forest regions. So, you know, the Southwest, Pacific Northwest is ideal for this. You know, not much in Hawaii. One case reported in Chicago. I don't know what that's about. Now worldwide, as you can see, you know, just a little bit in the United States, South America. Most of the regions where this happens is in Africa and Asia. Now, in Madagascar and sub-Saharan Africa, that accounts for 95% of all the reported cases worldwide. So you need to be careful when traveling to those areas for sure. Prevention. Prevention is pretty simple. Just avoid contact with rats and possible infected animals, and have your pets flea treated. I think it's like the third or fourth time I said that, but that's the best thing you can do for you and your pet. If you suspect you or someone has the plague, you need to go to the doctor right away, and you need to get on antibiotics. It's not something to play around with. You only have a few days before it gets worse, and if it ever gets to the pneumonic plague, you're pretty much dead. Vaccines, as I said earlier, there isn't anything readily available right now in the United States, but the military has contracted Denport to design a vaccine. It's currently in the research and development phase. If you want to know more about that, you can just Google Denport, and it talks a little bit about what the vaccine does. There's been talk about it when I was in the military and working for the military. It's not a secret or anything. But the main reason why they're developing it is so it protects us and soldiers against the weaponized version of the plague, because it's airborne and it can spread from human to human. So that's my presentation. Any questions, feel free to ask me. I look forward to answering everything you have. I have quite a bit of info, so ask away.